Okay, hi everyone. I'm going to give you a really brief recap of everything that we learned in this week's lecture, and I wanted to do it in a slightly different format using a different type of series of slides. But I did want to draw your attention back to what we had talked about earlier this week, which is about learning and memory, Eric Kandel's experiments, and how we use different neural circuits and the identification of different signaling molecules in these circuits to let us understand more about learning and memory. And so when we go back and we start reviewing things, I wanted to make sure that you knew about the different markers for the different neuronal compartments that I'll get to in a few moments, as well as protein kinase A, which was the very first molecule that was identified in this whole process. I then wanted to make the parallel between Eric Kandel's neural circuit model as well as the hippocampus, which we're more accustomed to looking at in terms of learning and memory. And then we'll talk very briefly about how we measure LTP in different contexts. So again, this goes back into the early 1960s and the late 1950s in terms of looking at different molecules that were associated with learning and memory. And we know that one individual that was responsible for this was James McConnell. He actually had created a really controversial model where different molecules that were associated with learning and memory, if they were given to naive animals, would actually allow them to retain that memory, suggesting that cannibalistic protein or RNA delivery into naive brains would actually allow for memories to be encoded in a unique fashion. And this actually put a brake on learning and memory investigations for a very long time because it was so controversial. It wasn't until much later that we started to go back and look at simplified models for what learning and memory actually encoded for.
So how much would we actually have to know from this slide?
So Sabina is asking a really good question, and the reason why I wanted you to know about this dissociated neuronal culture was to make sure that you understood that when Eric Kandel began his initial investigations, he was looking for a really easy model to begin working with. So you don't really need to know all of the details on this particular slide that you see here. For example, you don't need to know about the different days in vitro, but I do want you to make sure that you know that this dissociated neuronal culture is actually a really simplified type of synapse that most investigators would want to be using now. Hopefully that answers your questions.
It does, thanks. And coming to this slide, how much do we have to know from here?
Okay, another great question. And this is actually a slightly different type of question. So do you need to know that autopses represent a simplified synapse? Absolutely. So with dissociated cultures in that previous slide, as well as the autopses here, these would represent the simplified types of synapses that most investigators would use now. But I did point out that you should make sure that you know that from this autoptic culture here and the same one over here, that we can actually stain the same synapse with different markers like ProSAP1, which is specific for the postsynaptic compartment, as well as vGlut1 as a presynaptic marker, and then we can look at neuronal dendrites using the marker here. And all of them would be proteins that exist within that particular neuron. Hopefully that's going to be something that you're comfortable going back and learning.
Yes, thanks. Okay.
Okay, so this gets into what Eric Kandel was actually able to use. So he used Aplysia, and Aplysia was the simplest model that he could use at the time. Now, nowadays, we have many more diverse models like the dissociated cultures and autopses that would allow us to study changes in synaptic strength. But at the time, this was the best model that he could come up with. And what he decided to use was this simple procedural learning model that was associated with Aplysia in this gill withdrawal reflex. And this is this simple circuit that he decided that he wanted to examine.
So on this slide, would we actually have to, would this be a short answer type question, and how much detail would we have to know, or is it like a multiple type? So do we have to know from the beginning the steps, and I guess if we can bring that later with the L29.
So I think one of the things that you should realize is that this particular circuit that we see here on the right is a circuit that I want you to make sure that you know starts off with a sensory circuit, or a sensory neuron, and then it makes its way into a motor neuron, L7, which eventually causes the contraction of the gill muscle itself. The details here on the left-hand side aren't ones that you necessarily need to know.
Okay.
So we can actually look at it both in terms of the physiology by squirting water on the sensory neuron, or we can actually go in and use different electrodes to cause direct stimulation. The most important thing, if you were to note this, is that the presynaptic excitation is identical. It doesn't change, but we do see changes here on the postsynaptic side. So in other words, here's the first example that we're able to identify a change at the level of the synapse, where the synapse response is getting smaller and smaller over time. One of the other things is this model here.
Yes, this one has a lot of information on it. So what's important here? I guess the right side of the slide, or?
Actually, Sabina, I'm going to tell you that this is the same circuit that we looked at in that previous slide.
Right.
And here, what we're actually doing is taking another group of neurons and making sure that we understand that we can cause another form of learning that we call sensitization. And this is another form of non-associative learning. And you do need to know all the details that are found here on the left, but you also need to make sure that you check out all the information on the right, so that you know that serotonin, exactly, and that it's an interneuron that's involved, and that it involves a G protein coupled response in the postsynaptic cell that we're looking at.
Okay.
And again, just to reiterate, other things that you need to make sure that you're aware of on this particular series of slides is to make sure that you're spending the time to go over all of the molecular mechanisms that are involved, including the loss of activity of the protein channel once the G protein becomes activated. All the steps are outlined for you here on this particular diagram or on this particular slide, so please make sure that you're spending the time to go through all of them over the weekend or when you're going back to study. So ultimately, this is an invertebrate model system. It was a simplified model system that Eric Kandel used because he had none of the resources available to him at the time to do dissociated or autoptic cultures. Nowadays, though, because we're much more sophisticated and we have different types of models, this is the circuit that I'd like to, each of you to make sure that you are going over and spending some time on. Did you have some questions, Sabina?
This immediately looks like a lot of information. So again, is it from the beginning of the circuit that we have to know? Would this be a type of short answer question? And in that case, I guess we would have to know more detail, or?
So I'm going to tell you that you definitely need to know it from the beginning, the input coming in to the hippocampus. So this entire structure is the hippocampus, and you need to understand the pathway and the anatomy of the pathway, the perforant pathway, that brings sensory information in from the entorhinal cortex into the first cells that you see here, the granule cells of the dentate gyrus. Their axons in turn make up these mossy fibers, which then work their way over to CA3, and these in turn will send their axons together as a group known as the Schaffer collaterals to CA1. Okay. Now, the other important things that are pointed out to you is that the CA3 neurons actually don't require NMDA, and we'll get to that in a few moments, but that the CA1 neurons do use NMDA, and these are the sets of neurons and their synapses that we're the most interested in when we're taking a look at things. One other sort of hint for you as well, you should also make sure that you know the axons that leave this particular part of the hippocampus. I don't know if you remember those at all, Sabina?
Fimbriafornix?
It is, so the fimbriafornix, exactly. So make sure that you know the entire circuit as well as why information comes in through one pathway, the perforant, and leaves through the other, or the perforant pathway.
Okay.
So again, the other thing that you should make sure that you spend some time on, when we're looking at this particular Schaffer collateral and the CA1, we have three different ways that are outlined here that we can cause excitation. So we can use this first model, which is taking an electrode and using high 100 action potential per second stimulations, and we would then measure from the CA1 neuron, or we can use the same axon and then do theta bursts, where it's brief pulses at 100 hertz and measuring the responses that we see here. Some of the newer models that I'd like you to make sure that you're aware of include this model here known as STDP, which is a short form for spike timing dependent plasticity. And one of the things that happens here that's a little bit different is that we have an electrode here presynaptically as well as an electrode postsynaptically, and we stimulate both at the same time at very, very low levels. If everything works, this is the initial response, and after doing any of these different methods, whether it's 100 hertz or TBS or STDB, that same neuron will always show an increase, so we get a much bigger response than we had seen beforehand. So again, this is sort of the overall model. This is the baseline that we see, and then after tetanic stimulation of the Schaffer collaterals or STDP or theta burst stimulation, we will always see an increase. But this increase is actually really interesting. It starts off really high and then it becomes lower. And it turns out that a lot of this is actually due to the NMDA receptors. But before we get there, one of the things that we need to know is that big increase that we saw is known as induction, and then this plateau phase is known as maintenance. And we know that this NMDA receptor, by using specific blockers like MK801 or APV or AP5, is only involved in the induction, this upward phase here. But if we use the same blockers here after LTP has occurred, there is no effect on maintenance. So it's really important that you're able to distinguish that NMDA is important for the induction of LTP, but once you have LTP, the NMDA receptor doesn't become as important. So we sort of ended off here looking at some of the cellular and molecular pathways that are involved. And in the next lecture, we're actually going to go back and look at how NMDA receptors, through the influx of calcium, causes CAM kinase 2 as well as PKC to cause this LTP phenomenon, and how it's actually expressed. So I'm hoping that you'll spend this weekend kind of going over things, making sure that you're studying, and hopefully this 5-minute recap, along with the list that I gave you earlier, will help you in studying more effectively.